Hi, how are you,
Doing
Brianna?
doing well. Good to see you today.
Yes, we decided to do something a little different today, which is we get quite a few questions from our audiences, and we'd love to be responsive to what all of you are wondering about the aging space is. So this is the first of what I hope will be many Q&A sessions between the two of us.
Yeah, I have to say, I got the list of questions, and some of these questions, if we knew the answers, then you and I would be out of work already. So some of them are pretty big questions, but I'm sure we'll do our best to answer as many as we can, and highlight where there's still a lot more work to be done, and that's what you and I will be working on, I guess, the next few years here at the Buck. So why don't you let me kick off here with one of these questions? So why don't we start with perhaps the biggest one? With all of the different hallmarks and theories of aging, do you think that there is one single upstream cause of aging across all tissues, or could different tissues age for fundamentally different reasons? And that big old question is from Kyle.
That's actually a top question. Everyone loves simple mechanism, one thing that explains everything. And in some cases, it has happened. Sometimes science provides a model, a theory that just explains everything. Rarely, though, and certainly not in the case of aging. I'll speak about two different aspects. One is aging is pleomorphic. It is induced by a whole variety of mechanistic problems, what we call the hallmarks of aging. I do not believe there's a single cause of aging, and therefore I do not believe there will be a single treatment against aging. So I hope I'm not bursting anybody's bubble. You will hear some colleagues, tout their theories as if it were explaining everything, and we've heard about this: the epigenetic theory of aging, the stem cell theory of aging, oxidative stress theory of aging. I do think about aging as a pleomorphic problem with multiple issues that we will have to solve over the long term. So
I heard near Barzilai, I think it was, speak recently, and it was he or someone other, some other luminary in the space. And every time you go to an aging conference, there's that one slide with all of the hallmarks of aging placed in a circle, and everyone uses this slide. And they made the comment that it had to be a circle because nobody could be in the middle because they're all interconnected, and it's not possible to isolate one that isn't having some effects on the other. So I think that idea of geroscience, which was, of course, founded and conceived by scientists who were here at the Buck when it started 25 years ago, is that rather than just be one root cause, everything's interconnected, and rather than just have one treatment that will target one pathway that will, fix aging, where by the very nature of the problem, it's all interlinked, and modulating one pathway is going to benefit others. So I think I agree with you that it would be nice if there was a magic bullet and maybe one pathway would emerge as the dominant one. But based on what we can see now, it's too interconnected, too complicated to be kind of so simply distilled.
exactly, I totally agree with this. There's one other aspect where there's a unification process that's happening, which I think is important for the audience to know. And I'll make the analogy to what happened in the 1800s. People were dying mostly from infectious diseases. You had cholera, you had bubonic plague, all of these horrible diseases, pneumonia. People were essentially dying about 90% from infectious diseases. What happened in 1850 to 1900 is the realization there were microbes. And these microbes provided a unification because many of these diseases were driven by distinct microbes: tuberculosis, Vibrio cholera for cholera. And so there was something that came out of the work mostly of Pasteur, which was the germ theory of disease. And I think so that allowed people to really switch the way they were thinking of disease, first looking for an infectious agent, and then this led to penicillin, antibiotics, antivirals, and to essentially not the elimination, but disease of infectious disease origin.
A big inflection point in how we thought about it and deal with it.
And those diseases have mostly been eliminated, not completely, but they're certainly
Controlled.
not.
They don't kill as many people as they used
No,
to.
and now we've replaced this by a whole series of other diseases that we are looking in the same way: a heart attack, stroke, many forms of cancers, type 2 diabetes, and all of these diseases are driven by one single major factor, which is aging. And so this is something called the geroscience hypothesis, comes out of the Buck Institute's work. And I think my prediction is that 100 years from now, we will look back at this period as saying this is the time when they realized all of these diseases that were treated as independent occurrences, what I call whack-a-mole medicine, were actually driven by the aging process. So this is where I think the geroscience revolution is going to be taking place.
Yeah, I'd kind of love to take a time machine forward and look back at that now. I want to pull a bit more on this second piece of this question, which is about different tissues aging for different reasons. There seems like there could be something there. We know that there's, say, more protein misfolding, say, in the brain, and more, different types of changes that happen in the aging immune system. So, is there more you can say about, without there being one ring to rule them all as far as it goes with a hallmark that drives all of aging, are there processes that dominate in different tissues?
Absolutely. And I think we just talked about the idea that aging is a major risk factor for all of these diseases, but clearly not everybody is getting every one of these diseases. You see one person will get a heart attack, another one will get COPD, another one will get macular degeneration. And the reason for this is twofold. One is each organ has a series of frailty points. Obviously, if you're smoking, you're going to be causing local problems in the lung, so you'll be more likely to have lung cancer. So each organ has a unique set of frailty points that are influenced both by your genetics, and if you're high cholesterol genetically, or if you're eating a poor diet or not exercising. So that being said, now we're at the stage where we can actually measure aging in specific organs, and I think that's going to be allowing to determine for each individuals what are your frailty points, what should be watching for in your case. And that, in your case, is determined sometimes by lifestyle factors, some other times by genetic factors.
I think you had on this podcast series, we had a conversation with Tony Wiscori, and he's been developing organ-specific aging clocks based on blood measurements, right? Proteomic measurements that flag up which proteins might have come from and indicate damage or aging in specific organs. Does that relate to this kind of concept?
Absolutely. So you've all heard about these biomarkers, these clocks that allow you to measure aging. One big area of progress in the last couple of years, including from our own institute, David Furman has generated some new organ-specific clocks, but also the work of Tony Wiscori and his company that he started called Vero. I believe there's Steve Horvath and others, Vadim Gladyshev, are all looking at the same aspects. So how can we actually measure aging in individual organs? In each person, can we identify the really frailty points? And also, can we understand aging better? One of the things that came out of Tony's work that we discussed earlier is that when you look at individual organs and you try to determine which of these organs is more predictive of your demise, there are two organs that emerge, the immune system and the central nervous system, which makes sense. They're both distributed organs, so it means we should, most people, try to preserve immune aging and central nervous system aging. That being said, so here's another question from one of our audience, from Giovanni. So how should clinical trials measure success in slowing aging?
That's like the question that I wake up every morning thinking about and go to bed every night thinking about, because I think it's one of the most important questions that those of us who work in clinical geroscience are thinking about right now. And I want the listeners to know that it's not just us thinking about it here at the Buck. A week or so ago, so we were recording this in June, late May, I was over in DC at an FDA convention talking to regulators and industry experts about what would it look like. How would we know if a treatment or a drug slowed aging in humans? Because there is no regulatory pathway for that. As you mentioned, right now, aging is a risk factor for many diseases. The regulatory pathways are focused around getting approvals for a disease and not for the underlying aging biology. So this is a huge problem, but it's something that's being addressed currently. FDA are interested. The X Prize healthspan is putting in a big effort around communication and consensus building around endpoints that focus on muscle function, immune function, and cognitive health. And then also here at Buck and others across the US, we're participating in a program that ARPA-H is leading. ARPA-H is the government's health moonshot agency. So again, this is a big, audacious, ambitious project, trying to break new ground that hasn't been covered before. But the goal of this ARPA-H project is to get the FDA to agree to an indication for non-disease aging, healthy aging. How would we know, you're very well, Eric, you're very well, you still ride your bike, you still do hot Pilates, you're still, functioning really, really well. But how would we know, when things are starting to go awry, and could we intervene before you were not well to keep you functioning as well as you are now?
Actually, not so well today.
Not so well today.
You can hear from my voice, but
not so well today.
That's my immune system is not working so well.
And so really like the central concept here, which I think that the listeners need to, perk up their ears and listen about, because I hope that more people will hear more about this in the future, is this concept that the WHO developed called intrinsic capacity. So it was developed in 2015. It's been around a little while. The WHO has enough organizational heft still to really foreground this idea around the world. Intrinsic capacity is the sum of all of the capabilities available to you and me to live and enjoy our lives. So it has 5 subdomains: our physical function, can we move around? Our cognitive function, can we think and remember? Our psychological health, have we got the motivation and the social connections that are enriching our lives? Our sensory function, can we hear, can we see and engage with our environment? And lastly, our vitality, which is a little bit less specific, but it kind of speaks to our inflammatory and immune status, our muscle function, muscle and nutritional status, and really underpins those other kind of emergent properties. So ARPA-H and the FDA and all of the, all these bodies are starting to get together and think about, is intrinsic capacity something that we could measure as a main outcome in clinical studies? That means that the tests that we have to do in clinical studies, it won't just be a simple blood test, although biospecimens will almost definitely be involved. We'll actually have to use all of this new technology that we've got, maybe wearable devices, as well as microsampling devices so that people can test at home to really pull out much more granular detail about how people are functioning. So I think that at this moment, there's a regulatory interest in tailwind. There's a technology interest in tailwind with the wearables and our ability to analyze all of the data with AI and machine learning, as well as just, enough groundwork been done in the field by yourself, all our colleagues at the Buck, people who've been thinking about trial design for a while now, that potentially focusing on this retention of function, building an adult health curve. So we're all familiar with growth charts in pediatrics. You know where you stand relative to others at 6 months, at 12 months, at 18 months. Like, what if we had the same thing for everyone once they hit 40, say, and could follow people and say, well, you're in the top 90th percentile for 50-year-olds, or actually, no, you're in the bottom 20th percentile for 60-year-olds. Can we correct that? Could you then go to your doctor and they could give you some agent that's specifically targeted at geroscience to keep you aging better? So I hope that's where the future will go. It combines biomarkers with functional endpoints. Something I heard the FDA say a lot was they really care about how people feel, function, and survive. And I was kind of encouraged by the idea of feel and function because, quite frankly, if you're 70 and, you have a slightly high blood pressure, it's probably not something that you experience that much in your day-to-day life, and you could take a drug, it would lower it, but it's not going to make as much difference as if you take something that means that you can go up the stairs without being winded, or walk to the store that's half a mile away and you couldn't do that previously. So people want, when they take a drug that targets aging, for them to feel better soon. And so focusing on function, as well as bringing in all of these predictive biomarkers, I think is going to be, again, maybe something we look back on in 10, 15 years' time and wonder why we weren't doing it sooner.
You can already feel this shift happening, and I think it's an incredibly important one because, as you mentioned, what people care about is not having a low biological age.
Low CRP, who cares,
What people feel about, they want to be able to sit on the ground with their grandchildren. They want to be able to be mobile, to enjoy good food. So all of these things. And, both you and I are working with the super age game, which is another manifestation of this. Can we measure aging functionally in people? Can you actually train to maintain your mobility, your flexibility, and all this? So in some way, I think some of the X Prize competition is going to be driven by some of the same modules in the long term. So I think there's a lot of excitement in the field that we're coming into the age of really trying to change aging in a way that matters to people.
Yeah, and I think it's another thing that's an interesting trend that's hard to navigate is that with the increase in available data and our ability to process it, it feels like every other week there's some kind of new biomarker-based composite clock that predicts aging. And some of the things that I think about here, and from time to time I'll review papers and looking at what does it take to move something from an interesting result in a science paper to an actual biomarker that would be useful in a clinical trial. So think about what we see a lot with the epigenetic-based clocks. You can send off a blood sample and you've talked about this, and I know others have, that if you sent a blood sample from a different arm, separated by an hour, you might get something that's 5 or 10 or 15 years different. And I recently was reviewing a paper that talked about the effects of stress and the effects of meals on epigenetic clocks. And so it's really they're very useful and valuable. But if you need something to be stable enough to say, well, I went on, I went on an exercise program and I took a before and after epigenetic age. Is the difference that I'm seeing really due to that exercise program that I did, or is it some biological variation? So, these clocks and anything we use to measure success, we need to make sure we understand all of the caveats before they're proposed to be ready. And there's a difference as well between being kind of broadly useful in a longitudinal observational longitudinal cohort of like the UK Biobank, thousands and thousands of people. You can get enough signal in that because there's just so many people over so much time. But for you or me or individuals and smaller scale clinical trials, then there's an awful lot we need to think about before we pick, as powerful as a clock might be, a clock like that as an endpoint.
Great points.
Next question from the listeners that I think, again, ties nicely into what we were just talking about. So we've talked about what success would look like in clinical trials. We're not there yet, with having that framework, but we are doing a lot of work looking at interventions that extend lifespan in model organisms. We can pretty reliably make a worm or a mouse live longer in the lab. So we had a question from a listener that asked which interventions that are known to extend lifespan in model organisms will actually work in humans. So again, a big question. What do you think about that?
Well, I think if I had a crystal ball, I would tell you right now that the problem is the following. So we, as you mentioned, we have a whole series of interventions that have been documented to increase lifespan and health span in animal models. Probably the best known, that is the closest related to humans, would be a program run by the NIH called the ITP, Intervention Testing Program. So any scientist who works in the field can nominate drug candidates to the NIH to this program, and there's a 2-page submission that you can send in, and if they decide, okay, there's enough preliminary evidence in other animal models, in a worm, in fruit flies, then we'll test it in mice. And they do this across 3 different testing sites in the country. It's
same intervention.
Same intervention, sites. 3 different sites, so you eliminate That's sort of variation in the the type of mice and so on. And then they give the mice the compounds for the lifespan, and they measure aging in males and females. So they've gone through, I think, more than 70 different compounds. Out of those, I don't know the exact number, but it's close to 10 molecules have been shown to reproducibly increase lifespan across the 3 different cohorts. Interestingly, for reasons we don't clearly understand, there's a sexual dimorphism. That is, so we see a lot more increased lifespan in males than we do see in females. Not understand
across the board, which is super
across
interesting.
the board, which is interesting. We don't understand why that is. That being said, these molecules now, some of them, the most well known is rapamycin, but there are others that are drugs that actually we have in the clinic. Acarbose, it's a diabetes drug that prevents sugar absorption. There's So can we predict which one will actually work in humans? No. And this is where the biggest glut right now and sort of a bottleneck in the aging field lies, is that we have so many interventions that could go into humans. Problems is human trials are much more expensive, although here at the Buck Institute we are actively working at trying to, under your leadership, trying to make them cheaper, more effective, eventually decentralized. So I think there's a lot of area that this work needs to be done, unfortunately. And we know from previous experience in other animal model systems or other diseases that we can cure diabetes. We've done it 100 times in mice, and we still haven't done it in humans. So out of these 10 drugs, how many of them are going to make it into the clinic? I don't know. I hope at least a couple.
Right.
We'll see.
On the flip side as well, there are some things that don't show a lifespan effect in the ITP that are still leading candidates for gerotherapeutics in the clinic. And I was actually just at the Age annual meeting and talking to Sarah Espinosa, who's down at Cedars-Sinai and is one of the leading researchers running metformin trials with a geroscience kind of focus on her research. And she was saying that she'd been kind of discouraged because the ITP had shown metformin didn't have a lifespan effect. But I was there looking at this poster with all of this data, and it's like, it's really doing something. Metformin was one of the leading candidates for a gerotherapeutic trial not that long ago. And so, she and I kind of came to the conclusion that the ITP is one useful brick in the wall of data, but it doesn't mean we should throw the baby out with the bathwater insofar as if something hasn't worked in the ITP, it doesn't mean that it's not going to work in the clinic also, because of so many, just unavoidable differences between mouse and human biology and the difference between, doing an experiment in mice in a very controlled environment versus humans in this big, messy world. So, ITP, I think, is the gold standard, but it should always be interpreted relatively cautiously. And as you said, summarizing there at the end, until we have scalable and a unified vision of what success looks like, then it's hard to, make progress in this space. But we're getting there.
Totally agree. And I think, one key thing to realize about the ITP is looking mostly at lifespan.
Yes.
And so, and as you know, we're working on both healthspan and lifespan. So, and you could have a series of drugs that would actually really decrease morbidity and not increase maximal lifespan.
Something else came up at a meeting I was at recently, and I'm interested in your take. So, again, this FDA meeting, we're all talking about function, healthspan being really, really important. One of the speakers got on up and he said, well, the thing is, I hear you all talking about function, but if I killed you tomorrow, it wouldn't matter, whether or not you were going to be functional. So centrally, we still do care about lifespan. His central thesis was that mortality, not lifespan per se, but mortality rate should be the thing that we look at. And I think that from other people that I speak to in the field, the feeling is that mortality just means the trials have to be too big, because especially in younger people, you have to follow people, the cohort rate. I can see why, from a practical perspective, mortality is not attractive. Where do you stand on, well, at the end of the day, it matters, the toes test. Jamie Justice says, toes up, toes down, like whether you're alive, whether you're dead, that's what matters most. Where do you sit on that?
I sit on that based on my personal experience talking to audiences, and I ask quite often the question, who wants to live to 120? This is like a long, long life. Typical audience, I will have 1 out of 100 people raise their hands. If I rephrase the question and say, who wants to live to 120 in the same shape as what you would expect to be at 60, surrounded by your family, with your job and all this, 99% of people. So everybody wants to live long. Nobody wants to live sick.
Right.
And I think in some way, quite often the message that I'm giving is misunderstood as to being a healthspan message. No, it's long life, but not at all costs.
They're not mutually exclusive,
No.
right? They are codependent on one another. Yeah, no, I agree. It was an interesting, very provocative. The speaker was very provocative, I think deliberately, because of the other points that had been raised. But it made me kind of reevaluate my own thinking a bit. So we'll move on to the next question. I think that hopefully listeners can tell that you and I are both really excited about all of the growth in the field, the opportunities in the field. And so one of the questions is about young scientists getting into the field. And our listener asked, how can young scientists who want to work in translational aging research or start biotech companies get their foot in the door? What path best prepares someone to develop therapies for targeting aging? And I think you and I both probably have some thoughts about this, but why don't you kick us off?
Yeah, there is not such a thing as a path. And that's probably, if looking at both of us, maybe we can share our examples because we are in some way completely unorthodox ways to get into aging research. I was trained as a physician. I did my whole research, my career mostly in research. We discovered by chance a gene and a protein that were involved in the aging pathways, and this is how I got into aging. I just thought this is...
And you don't have a PhD.
I don't have a PhD. No, I have an MD and a long postdoc. So that,
a lifelong postdoc.
Exactly. So this is... so for me, I tell people, the biggest driver, you need some form of education, number one. We'll hear about what you went through, which is completely separate from what I did, which is equally successful. So you need passion, you need interest, you need some training at some level. So that would be the key factor.
Before I say about my own journey, that's a prompt for me to say that Dr. Newman, who's here at the Buck, one of our colleagues here at the Buck, as well as others at UConn, and I'm trying to think which other institutes participate, but the NIH recently funded a consortium called the Geroscience Education and Training Consortium, GET, and they have been developing over the last 2 or 3 years an actual educational program for people who want to, who have come from other areas of biomedical research, but who want to learn about aging biology and aging research. So if anyone's a trainee and is listening to this, they should go away and look up the GET network, look at the educational opportunities that are coming online. I believe there'll be some online courses that are going to be offered through various institutions, and that's the one resource that I definitely remember, but I'm sure that they're developing other resources as well. And that would be a really great place to start for someone whose PhD is related to aging, perhaps not purely in the aging field, or who wants to learn about aging to start a company. But back to where I came from, I guess. Similar to you, wanted to impact people's health and wellness, started medical school over at Oxford University. Halfway through my medical training, got into research, firstly as a participant, felt like it was kind of fun. It also gave me time to pursue my rowing, which was, my side hustle as a professional athlete, and studying my PhD at Oxford. Following my PhD, I actually moved over to the US to join a company. So I didn't do a traditional postdoc at all. I moved into a company. I felt like getting experience in a startup. There is a couple of ways that you can get exposed to the types of skills that you're going to need to start a company. You can either go and work in a really small company like I did, where you are one of 10 people and you're really in the room when all of those hard decisions are being made. You get a lot more exposure. Or you can go and be in a pharmaceutical company and work through, often they'll have formal training programs for people, recent graduates or PhD graduates, and there you'll have more structure. You'll get a higher, different kind of exposure. And so that's 2 ways you could think about ways to train post education. For me, I was in the company, got a lot of exposure that way, and then ended up at the Buck kind of by a happy accident, because I was working on a research area that you were also working on.
It was not a happy accident. We've recruited you very actively.
Very actively. Okay. I felt it was, it was great. I'm very happy to be here. So yeah, I think being in a startup, going into pharmaceutical companies, at the meeting I was just at, there was people who had done both, and I think one message that resonates with me when I look at my career, and this panel that I listened to also said, is that there's never too late to change. You can go and be in pharma for 5 years, learn a load, and then go and join a biotech, or you can even go from being in a biotech back into a postdoc if you wanted to. So I think there's a lot of pressure on people to make a decision and make the right one instantly. And you were thinking you were going to be a physician. I never thought that I would be here doing this. So I think if people can take away one thing, it's follow your passion, follow your interests. Don't be afraid to try things, because mostly there's not that many one-way doors that you can't, change courses further on down the line. That
being said, we're both incredibly lucky to be in this job, in our respective jobs here at the Buck. And there is a bit of a dark cloud surrounding all of us in biomedical research at this time, which I hope will resolve in the near future, which is the NIH and its role in funding the whole effort is a little bit is being challenged right now. So for those of you who are out there, who've already gone through some training and looking for a job in the future, I would say I try to stay optimistic because I think the work that we do is so important. The fund will come. We're learning how to navigate a different system. And in the case here at the BUC, we've been able to attract significant funding from ARPA-H. So the work is actually, we had our biggest year last year. So I would say let's not, I try not to let the gloom and doom affect us. I think be creative. Network, talk to us, talk to the people in the field. There's still a lot of very good job right now, and there will be more in the future. I'm really confident that if you're considering doing a career in biomedical research, of all the fields that you could go into, aging research is the one to go into.
I'd agree. Okay, I think we have time for maybe one more question before we have to wrap up this session. Although I would say this has been really fun. So if anyone has further questions, just send them in to us and we'll record another of these episodes sometime in the future.
I agree. Love those.
Been very fun. So to close us out, I know you've been thinking a lot, Eric, about science communication. And we had a question came in that is... I think captures a lot of the sentiment nowadays. In a world that is saturated with health influencers, endless information sources, AI tools, and both credible and questionable experts, how should someone figure out what is actually reliable? And I think we all feel that in every element of our lives. So how have you been thinking about building credibility, weeding out less credible sources in your own internet bathing?
Yes. Yes, I'm both a practicing, physician and scientist in the field, like you are, but also I'm a consumer. So I'm on Instagram and I get these reels, touting this supplement, and I must recognize from time to time I fall for it. I'll buy something and it comes into the mail. I'm realizing, why did I buy this? And that's attention, which I think is both necessary and something to be aware of. So one aspect you mentioned that I'm passionate about, communication, and that really is linked to this whole question. We live in an environment where everybody can actually communicate. We used to have a very rigid structure by which information was being communicated and was almost implicitly trusted. That has changed. Anybody who has a telephone and an Instagram account can become an influencer. If you're good enough, you can actually make a good living doing this. This did not exist 15 years ago. There are people...
didn't exist probably like 5 years ago,
exactly.
That you could be a kid and be like, Mom and Dad, I want to be an influencer when I grow up. Yeah. And like, as you say, it's like a career.
It's a career. So it's not only bad, I think it is actually an incredible democratization of the ability to share information and so on. In our world, it can be problematic because there is real progress being made in aging research, but there's a lot of quackery. Snake oil is rampant. This used to be called Dr. Google. Right now, I call it Instagram medicine. This is the idea that you're relying for making some medical decisions by someone who sits in his garage and sells you a pill that he's been making in his garage,
or sits in his multi-million-dollar mansion.
Exactly, actually, in Bali, probably.
Yeah.
So. That being said, I think one of the problems is, as physicians, as scientists, we've been reluctant to enter that debate. And I think we need to be much more, because we haven't done this historically, and we're also passionate about our science, we forget to communicate it. And I think it is something critical for us to engage in. So you might have seen that I'm much more active on Instagram, on LinkedIn. We now have a Substack. The reason for this is not for my personal self-aggrandizement. It's more the idea that we need to bring our voice to the public. And the listener is asking, how do you decide who to listen to? I would say, listen to us. We don't have all the answers, but we're grounded in science. The Buck Institute is a recognized, 26-year-old organization that's focused on science. 300 scientists working here. I mean, we're not the only ones, obviously, but if you're going to be getting some of your information, at least listen to us and then listen to others. We have not only my social media accounts, but also the Buck itself. We have this podcast. We have a newsletter. There's many ways in which you can engage in this community. And from there, migrate into other people who are a recognized source of authority. And, be aware, be questioning of everything you hear on the internet. There's a lot of craziness right now. AI has just compounded this to some degree because you can now make fake videos. So we're entering an era where, because of AI largely, a lot of the information that you're going to be subjected to is going to be fake.
I do feel, though, that change in the last year has maybe increased people's willingness to be skeptical. Or at least made it like this huge, you look at something and you think, every time I see something on the internet now, I ask myself, is this AI or not? And so I think that building in that reflex and that skill of asking and then knowing how to, look at the provenance of something, being forced to the foreground a bit more by AI could be helpful for the whole space. But the problem is it's work. It's work to assimilate, cross-check against a few trusted sources, and, go do that digging and look and see. So, and I think that's in direct opposition to how that information is meant to be consumed. So I think how we make conclusions from information probably has to change as there's more information, a higher risk that it's misinformation and not information. We have to build good habits and skills around, building our own conclusions. It's funny because I think you consume social media to sort of switch your brain off, and I think maybe this is a call to action that you actually need to switch your brain back on when you're using these platforms before you make any life choices based on what you read there.
I agree. That's the place to be the most skeptical, and at the same time, I think it is here to stay with us. It is part of what I call the empowered patient, the idea that all of us are becoming participant to our medical decisions. So versus having a doctor giving you orders, I think all of us, will become much more active in the decisions that lead to this treatment or this supplement. Just be especially critical or suspicious of people who claim to have all of the answers. Because the bottom line is that science is, as one incredible virtue, is that it makes mistakes and it recognizes them. So if you hear about something that just sounds too good to be true, that should be a reflex. Well, maybe there's not so good to be true. Someone is trying to sell me onto something. And we are, I fall for this. It always amazes me from time to time. I'll fall for something and then I start digging around. something just sounds too good to be true, claiming from a paper, and I'll go look in the paper. And it's mind-blowing how people will just distort what they're reading for the purpose of making some noise.
That really scares me because I know that as a scientist, I'm a PhD, I'm a research assistant professor. This is my job. And if it takes me to follow a link to PubMed, go into a paper, and then go, into something, and actually make a conclusion based on all of those years of expertise, how is anyone else meant to, to do that? So I think it's fair enough that people feel a little overwhelmed by this, because I feel overwhelmed by that.
And I agree. And I am amazed by the amount of sort of manipulation of existing paper that some people will go through. So again, this brings us back, rather than trust all these disparate sources that you do not know anything about. As I mentioned, the guy in his garage selling a supplement versus a place like the Buck Institute. I would argue that many people should migrate back towards some of these trusted sources. Now, I have been told, while your Buck are so conservative, that's not what I want to hear. I want to hear about someone who's going to tell me I'm going to live to 150. I said, well, there you go. You can either live in that illusion that you're going to live to 150, or you can say, well, aging is really complicated. The Buck is studying it with 300 people, $70 million a year, and they've come to the conclusion that the 150-year-old is probably not going to happen soon. So decide who you want to trust. This is really You can either dream or you can really look at the hard data.
Yeah, I totally agree. I'm really proud to be a member of the community here at the Buck. And I say to the listeners, all of the people that we bring on and we have conversations with, those are also people who we trust. And so I'd encourage all the listeners to look up our guests. Those are also great sources of information. And you listeners, by consuming this podcast, by being part of our community, you're getting a better shot at getting trusted information and learning who those trusted partners are. So thank you for being on this journey with us.
Thank you all. I think it's great to have you as an active audience, and just keep sending those questions in. We love to read them, and we love even better to answer them.
Thanks for sitting down today, Eric.
Thank you.
Thank you so much for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts.
We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to us here directing the recordings, and the esteemed Sharif Azad weaves the show together for you.
If you're listening to this podcast, you know that there has never been a more exciting time in research on aging. Discoveries in the labs are moving into the clinic to help us all live better longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate. This episode was sponsored by Ashton Thomas Private Wealth, where discipline shapes vision and vision builds legacy. Learn more at ashtonthomaspw.com.
Investment advisory services are provided by Ashton Thomas Private Wealth, LLC, and Ashton Thomas Securities, LLC, SEC-registered investment advisors. Securities are offered through Ashton Thomas Securities, LLC, a registered broker-dealer and member of FINRA SIPC.